Okay, so my name is Kristoffer Sahlin, and I'm a postdoc in Paul Medvedev's lab. And I can see that he's not here now, so yeah, but that's good anyway, because I won't be talking about our work since it's fairly recent. So I'm just going to talk about some of the work I did during my PhD. So there I worked mainly with modeling read pair data for applications such as scaffolding of genome assemblies and structural variation detection. So to go through some definitions. So what I mean with a read pair is basically two reads sequenced from the ends of a fragment. And this, okay, I'll skip that one.
You have the manual for
Oh, okay. So read sequence from the ends of a fragment, and they're separated with some unknown distance that's usually referred to as insert size, and I denote this insert size with the variable x. And some things to realize about this is that when you get all these reads from a sequencing project, there will be some natural variation of insert size within a read library. So here's an example showing insert size distribution of all insert sizes of all the read pairs from a distribution. And this looks very normal, but this does not have to be the case at all. It could be, yeah, usually it has thicker tails, for instance. So denote the insert size with x, and this distribution of insert sizes as f. And these read pairs can come from sequencing technologies such as illumina paired end reads, illumina mate pair reads, Foss-Mitter backends. It's just different ways to generate them. And these read pairs can be utilized in many different applications, such as scaffolding and structural variation detection. So if the two reads within a read pair maps to two different contigs, they give some information on the distance, orientation, and order that each contigs have with respect to each other. And in structural variation detection, if you map down the two reads within a read pair onto the reference genome, so this is donor read pair sequenced from an individual. If you map them down, if they map to the approximate distance that you expect, they don't signal that any variant is present. If they map close to each other, they might signal that an insertion is present by this red segment here. And if they map too far from each other, they might signal that the deletion is present in the donor genome. So that's some applications. So a common assumption when working with these read pairs has been that for any given application, say that we do scaffolding, so the links span two contigs, they are drawn from this distribution, and that's fairly intuitive to say, like, yeah, but if these are all the insert sizes generated from a sequencing run, of course, the observations we observe spanning two contigs, they will be from this distribution. Similarly, in variation detection, all the read pairs spanning this position, for instance, if we're testing for insertion or deletion in this position, all the read pairs from this spanning this position will also be drawn from this distribution. So that's a common assumption. And here, for instance, you could sort of extend further and say that, okay, the average insert size in any of these applications should equal to the mean of this library, which I denote with mu here. So mu would be 2000 in this case, for instance. So common assumption. And now this is the most important slide. So if you want to remember anything from this presentation, it's this slide. So this is the key idea of our work, is basically this common assumption is not true. So reads are not drawn from that initial distribution when you look at the specific subset of reads. And what I mean with that is that a subset of read is, for instance, all the reads spanning a certain position, or linking to contigs, or spanning a certain region. So, so this can be seen fairly intuitively by just looking. So I've color-coded this insert size distribution. This is the full of all insert sizes from a sequencing project. Shorter ones are in red, greener ones, they are the larger ones, they're in green, so color-coded. And if you just sort the reads with respect to the leftmost read on how they map onto a genome, and you just fix any position P on the genome, you can sort of, it's intuitively to see that, yeah, but these green guys, they have larger, higher span coverage, so they will be more abundant over any given position. So as you can see here, the red ones fail to span while the green ones start to span earlier. Similarly for contact scaffolding, the red ones doesn't make it over, but the green guys span over. So something needs to be done with the different length. They need to be weighted, basically. And this is just like the main principle. And then everything is just, from now on, it's just details and some results and random stuff. So for instance, I'll just show you a very simple model in the very simplest case, which is, let's say we want to model this. What is the distribution of insert sizes over a given position? So this is a copy-pasted formula. This you can just ignore this. This is the probability of insert sizes over a position. We claim that it's proportional to, first of all, the probability of drawing it from the initial insert size distribution itself, times some weight function, and you can make that how complicated you want with illumina GC bias, insert size length, and so on. But in just the very simplest case, we just say, yeah, but we can just count the number of possible placements that each insert size has over a position. And this is basically just so this given insert size has some certain possible placement. It can span this position, which is the insert size itself. It has X many placements. So in this simple case, we just have that the true insert size distribution over a position is just X times the full distribution itself. Proportional, yeah. Okay, proportional. It's a weighted, so yeah. And
So you have to renormalize it to be a distribution.
yeah, exactly. So it's proportional, and so we do not require any normality, or this is just for intuition, and it's pretty cool that in case f is normally distributed with these parameters, you can get approximate analytical formulas, which is like just nice if you're a mathematician and you want to like derive something. So so the position specific mean over any position is the mean of the global library of the total insert size library plus something positive that's dependent on the variance of the library itself. And it's fairly intuitive that if you have larger discrepancy between the larger ones and the smaller ones, the larger ones will be more abundant. Similarly, the position-specific standard deviation under this assumption is the standard deviation times something that's less than 1, so it's always smaller. One way this can be used is then, so in a very simple case, common thing when testing for variance with read pairs over a position, you say, yeah, but let's look at average insert size over a position. Let's see if it's significantly different from the mean, and then we can sort of get some standard normal test and just see if it's significant deviation and score if it's a variant. So we basically propose, so this has been done in some tools, and we just say like, yeah, let's just switch this out under a very simple assumption and just test it against the position specific and use the position specific variance and mean. And so this is an example from biological data. So we have a mate pair library. You can see here, so it's not normal at all, more like a log normal distribution here, I think. And we just looked at human chromosome 13. And what you can see is that the mode is around 2,000, the empirical mean is around 2,600 for this library. However, if we plot the average insert size over all the 88 million mappable positions on the genome, you see that they cluster tightly around 3,700, so around 1,000 base pair more than the empirical mean of this distribution. And if we just do the standard normal test and get p-values of insert size of variance, like this test that I just described in the previous slide, you can see that the p-value distribution is basically all positions are significant under this model. In our case, we get much more uniform distribution, which is, you want a uniform given that your data is generated under the null hypothesis. However, you see some enrichment still under significant p-values. It's a combination of things. First of all, this is not data generated from the null hypothesis. This is the real data, so some of these are true positives. There will be real variance. Some of it is due to GC bias. There are also some other artifacts with illumina mate pair libraries, but the main take-home message is basically the difference between these two plots. So just in summary, so I only described this in a very the simplest most case, but we did some modeling of this also within scaffolding and so on. And so the scaffolding part we published in 2012 already, so that was the first thing I published, and that basically showed that you improve gap size estimation if you want to do scaffolding. This claim is this is something I claim now. We didn't do it in the paper, but in scaffolding, if you can formulate, if your constraints to an optimization problem, like your linear constraints, is better estimated, then I hope that you can also get better solutions. But this is not something that we support with any data. We haven't checked into the significance of this, but it should be intuitive. And the structural variation detection part that I talked about more today, that was presented in Recom this year, and it's accepted to Journal of Computational Biology. And there we show more or less, a result of this that I've shown is that it increases power in detecting insertions when using read pairs. And it removes lots of false positive deletion calls. And this is basically because of the biased testing. However, as for any statistical approach, it will remove some true positives, but the ratio is fairly favorable. Yeah, and that has been one of the main things, that deletions are much easier to detect than insertions. And that's true also still for many reasons, but maybe it doesn't have to be that hard to detect insertions and reduce the that's the main idea with this work. There are still some challenges with our super simple models of GC bias in Illumina. There's even some worse things, that similar insert size sometimes tend to cluster over regions, which is nasty. But overall, we show the significant improvement. So now coming to Postlab, we still investigate some methods for structural variation detection and assembly using newer technologies to generate reads, for instance 10x genomics and TrueSiC. So it's basically barcoded illumina reads that can increase the power in detection, structural variation detection. We also look at PacBio genomic and transcriptomic data, as you heard a presentation about the PacBio transcriptome data yesterday. My main interests are similar to you guys, statistics, both the modeling and inference part, and probabilistic graphical models, and all kinds of algorithms to infer them. And some acknowledgements then. So people involved in this work has been Nathaniel Street, Matthias Frånberg, running experiments and so on, and Lars Arvestad, which was my PhD supervisor. And I would, of course, like to thank Paul, although he's not here, for really yeah, no, but he's great. You should talk to him about project management and see what's hot, and because he can steer you into the right direction and stuff. So I'm really thankful for that. He has been really helpful. Yeah, thank you.